So now let's get into a little bit more detail in those phases. So this slide is showing you a summary of phases prophase 1, metaphase 1, anaphase 1, and telophase 1. So in essence, a summary of meiosis 1. At the top, we can see actual photographs through microscopy of the genetic material and undergoing the different phases of meiosis 1. And at the bottom, we see the artist's rendition of the same process. So if you've studied mitosis or the last chapter, notice a lot of things are very similar. We have the centrosome with the centrioles. The centrioles form spindle fibers and they reach the opposite sides of the cell, and they're going to end up pulling the genetic material apart. And then notice at the end we've reformed our nuclear envelope and cytokinesis has occurred and we've separated the cells. So what I like to call the background information is the same. No matter whether you're prophase of mitosis or meiosis, you're going to have the genetic material condensing and the centrioles moving and forming spindle apparati. In metaphase, you're going to have the genetic material aligned at the center of the cell with the spindle apparatus holding on to it. At anaphase, you're going to have the spindle fiber shortening, pulling the genetic material apart. At telophase, you form the two clusters of your nuclear material, and nuclear envelopes form around them, and then cytokinesis can occur. So that background information is the same whether you're in mitosis, meiosis 1, or meiosis 2. What is unique is what's happening to the genetic material. If you recall, in mitosis, we did not have homologous chromosomes aligning themselves it's called synapsis and being held together. Here we do. So notice we have these short genetic material from the mother that has replicated. So this is one chromosome, and underneath of it we can see the same genetic material from the father. So these homologous chromosomes have come together, and that process is called synapsis. Same thing has occurred here. But notice we have a swapping at the ends of the genetic material in this long set of chromosomes. That is called crossing over, and that ensures genetic variability. And notice the crossing over event happened in prophase 1, and it stayed through metaphase 1. It became evident in anaphase 1. When we separate them apart, we can clearly see the swapping that has occurred, and then it continues through telophase 1. So back in prophase 1, if a crossing over event has occurred, which is the swapping of genetic material between homologous chromosomes, we have increased our genetic variability. Also notice from the parent cell that we started with to the two daughter cells, we are reducing our genetic material by half. So that's why up here it says we're going from one diploid nucleus, this is a diploid nucleus, to two haploid nuclei. Each one of these are haploid, or they contain half the genetic material. So if we count 1, 2, 3, 4, 1, 2, 1, 2. So we have achieved the haploid state at the end of meiosis 1. So then you might ask, well, why divide again if we're already haploid? Because even though we're already haploid here, each chromosome is made up of two sister chromatids, and we need each chromosome to be made up of only one chromatid. So meiosis 2 must occur. Notice again in meiosis 2, all the names of the phases are the same. It's just now we have a Roman numeral 2 after them. Notice as well all of what I like to call the background information is occurring. The genetic material aligning towards the center or the equator of the cell, the centrioles at the poles forming spindle apparatus, the genetic material being pulled apart in anaphase 2, and then in telophase 2 with cytokinesis following, we have our nuclei forming and our cells splitting in half. Notice what's unique though between meiosis 1 and meiosis 2 is now we have two cells that this process is occurring with. No DNA replication has occurred. Synthesis of DNA does not occur. We go back to where we started with, and that's going backwards. So DNA synthesis does not occur. We take our genetic material, which is already haploid, but each chromosome is made up of two chromatids, and we go through all of the steps of division until we end up with each nucleus now being made up of chromosomes that are only made up of one chromatid. So again, if we go back here, this chromosome has two chromatids. This chromosome has two, so it's haploid. It's two chromosomes, but each chromosome has two chromatids. Here, where each chromosome is one chromatid, and we are haploid. And we're basically done. And notice, if we go back one slide, we started with one cell, and we end up, that's diploid. We started with one cell that's diploid, and we end up with 4 cells, each one being haploid. And within those 4 cells, we have great genetic variety. Obviously, they don't have the same number or amount of genetic material as the parent cell. Additionally, they're not identical to each other. So all of these are haploid cells, but notice in this one, this is the only one where we have the one long blue and a short red. Here we have a short red, but our long blue is also part red. Here we have a short blue, like we have here, but in this one, our long chromosome is mostly pink, a little bit of blue, and here it's completely pink. Again, the colors are representing where the chromosomes came from, and the color just is a simplified way of showing the genetic variability that's occurring. So let's kind of focus in on this crossing over event. Here we have a chromosome made up of 2 chromatids, and the letters are representing alleles, which are molecular forms of the genes. And most people are aware that in essence, for each trait, you have a dominant gene and a recessive gene. Dominant genes are represented by capital letters, recessive genes are represented by lowercase letters. So this father's chromosome that has undergone DNA replication and is now made up of 2 sister chromatids, we're focusing in on 2 genes, and he is contributing the dominance of those 2 genes, whereas the mother contributed the recessives. If a crossing over event had occurred, so here the maternal and paternal chromosomes have synapsed, they've aligned themselves so that they can exchange genetic material. Here they're exchanging that genetic material, and they're basically going to swap portions of DNA. The end result is the chromosomes from the father are no longer containing the dominant genes for both A and B. The B is now a recessive gene that came from the mother, and the dominant B gene that was from the father is now on the chromosome from the mother. So this is a crossing over event, which is in essence a swapping of the genetic material, which again increases genetic variability. So here we have our cell with the paternal genetic material and the maternal genetic material. The maternal genetic material is a chromosome made up of 2 chromatids. Same thing here with the paternal. And here we can see that the genetic material has synapsed or aligned. This latter is what's called a synaptonemal complex. And this protein lattice work ensures that the paternal and maternal chromosomes align themselves so that when they swap genes, they are swapping intact genes. They're not, like splitting a gene in half, which would mean that we'd have too much genetic material on one and too little on another. That can happen, but it's not very common. And by the way, they estimate that 3 to 5 crossing over events occur per homologous chromosome.